So now we're on to part 4 of our lecture on photosynthesis and the light reactions here. We're going to try and finish up. So we have the first step in the light reaction, again being the excitation of the electrons in photosystem 2, which is also called the P680 photosystem. And you can see here we have electrons being excited to a higher level, and then they are accepted by that primary acceptor molecule within the reaction center of the photosystem. The electrons that have been passed up have to be replaced, and so those are going to be replaced by the splitting of water. So water, again, is going to donate 2 electrons, and those electrons are going to be from our hydrogens, so we end up with 2 hydrogen protons and a single oxygen molecule, which is going to bind with the other oxygen molecule in order to become an O2 gas molecule. And this is just showing that water molecule again being split with its 1 oxygen and its 2 hydrogens. The oxygen joining with another oxygen molecule to become the gas that will escape through the pores in the bottom of the leaf of our plant. Those hydrogen protons, just to remind you, are going to end up in the thylakoid space in order to make and generate a chemiosmotic gradient that will be used by that ATP synthase pump in order to phosphorylate ADP to ATP. So once those electrons are passed up to that primary acceptor, the primary acceptor protein is going to pass them along through a series of reactions. And those reactions are called the electron transport chain. And one of the large molecules that they're going to pass through is called the cytochrome complex. This is all part of the first electron transport chain in the light reactions. So you can see just the process here of electrons being passed along. And then one of the side benefits of this electron transport chain in the first half of the light reactions is that they are providing an opportunity for hydrogen protons to also pass into the thylakoid space to increase that proton concentration on the inside there. And then that's a picture of the ATP synthase, which acts as a pump essentially to phosphorylate ADP to ATP. That ATP that's produced is then going to be used by the plant to generate energy for our Calvin cycle reactions. And it's also going to be used by the plant in other areas, other functions within the cell. So now we've gone through the first electron transport chain, and our electron has lost a lot of energy along the way, or our pair of electrons actually. And so those electrons arrive at photosystem 1, which is also called the P700 photosystem. And again, that says chlorophyll B there, but it actually is chlorophyll A. So these slides need to get edited, but I want you to be clear that we now believe that both of these photosystems are filled with pigments, chlorophyll A. So the sunlight, wavelengths from the sunlight in the form of photons are used by photosystem 1 to excite electrons that are present in the chlorophyll A molecules to again start this electron transport chain process a second time. So we're going to see the same thing happen here where the electrons get excited and they're passed up to a higher primary acceptor state. And then those electrons that have to be replaced by the chlorophyll molecules are replaced from that first electron transport chain that we have going on right here. So this is going to provide electrons to replace those that have been raised and put on this primary acceptor molecule by the energy of the sun. Finally, this is the second electron transport chain, and what the major molecule that we have being used in this chain is ferredoxin here. And ferredoxin is going to eventually pass the electrons on to the NADP ion right here, which is missing an electron already, so it does need a pair of electrons. So it needs a pair of electrons and 2 hydrogen protons in order to make the complete stable molecule NADPH, which will now become the electron transporter for the Kelvin cycle. So you can think of the light reaction as actually storing electrons into the bank, and the bank is going to then be used in the Kelvin cycle in the light independent reactions. And this is the summary of all that we've just been going through here. So you can see each of the steps. And now we're ready to look at the Kelvin cycle. But before we do that, I want to stop and look at the ATP synthase and how this proton gradient allows the transformation of ADP to ATP. So, and also this same mechanism is used in cellular respiration, which we'll look at in our next chapter. But this is comparing the two and showing that these are the things that we have in common between the two processes. So the energy from photosynthesis is going to be sunlight, and the energy in respiration is going to be the breakdown of glucose molecules. And that energy is then used to move the electrons along a electron transport chain. And then there's going to be a proton gradient, which means a high concentration of protons on one side of a membrane, which will be used to generate a pump in the ATP synthase pump. And this synthase pump actually turns like a little motor to actually pump these protons out into the stroma in the chloroplast. And that flow of protons bonds the inorganic phosphate here to ADP, producing our ATP. And this is just a summary again. Something you can stop and write down if you'd like to add to your notes. But this is a summary of the electron transport chain in photosynthesis. There's one thing before we go on to the Kelvin cycle that we need to stop and take a look at, and that is what we call non-cyclic photophosphorylation. So there's something that can occur in this process that stops the electrons from going on to the NADP molecules. So that final electron carrier molecule doesn't get the electrons sometimes, because sometimes there are conditions in the cell that cause this cyclic photophosphorylation to occur. So what happens is the electrons do get elevated in the first two steps between photosystem 2 and photosystem 1. But if the photosystem 1 can't pass its electrons on to the NADP molecule, it'll cycle those electrons back to photosystem 2. And it actually cycles them back to the cytochrome complex that is in the first electron transport chain. And so what happens in that first electron transport chain is we get a lot more of the hydrogen protons being brought into the thylakoid space. So in that way, indirectly, this cyclic photophosphorylation allows the chloroplasts to produce more ATP. The only problem is it will not be able to produce NADPH, which will not allow the Calvin cycle to then run and make carbohydrates. But if for some reason the plant needs a sudden burst of ATP as an energy source, then this is a process that would be very helpful because it's going to produce a lot more ATP in a short amount of time. So another thing to keep in mind is in order to make just one molecule of glucose, we need 18 ATP molecules and 12 NADPH molecules. So just one splitting of water molecule and the passing of a pair of electrons is only going to produce one NADPH. So we actually have to run that electron transport chain at a pretty quick pace in order to just even make one molecule of glucose. And this is just another diagram showing you the photophosphorylation. So non-cyclic photophosphorylation means that it actually continues on. If it's non-cyclic, it's not going in a circle, it's not circling back, so it's keeping it on its linear path. So it's actually going from photosystem 2 down to photosystem 1, and then right on, continuing in that linear fashion to the NADPH molecule. So now we have NADPH stored away and ready for use in the Calvin cycle. But with cyclic photophosphorylation, we actually are not going to end up going all the way forward in this linear fashion. We're actually going to circle back. So you can see here we're looking the same as non-cyclic, but here we have that circling back occurring. So we never get down into this second electron transport chain. It just cycles back in a circle back to the cytochrome complex. And then again we're going to end up with a lot more ADP. So this is our summary of photosynthesis so far, and these are questions that you should be able to answer by reading through your textbook and by listening to this and reviewing this PowerPoint. And so pause this if you need to, and make sure you can answer each of these questions. So next we're going to move on to the Calvin cycle, or the light independent reactions. And right, this is the wrong Calvin. It's actually named after a man whose last name was Kelvin. And just to review, our light reactions converted solar energy to chemical energy. And we were able to produce NADPH, which is going to be our electron transport molecule into our Calvin cycle. And our NADPH is going to be the source of our reducing power and able to run our reduction reactions. So now what we were looking at with our light reactions was the photo part of photosynthesis. Now we want to take a look at the synthesis part of photosynthesis, the actual building of things. So synthesis